Okay, let's talk AP Bio, specifically like the whole DNA unit. Specifically, we're going to talk about the DNA structure right now to start. So what I have right here is the DNA like structure. It consists of a deoxyribose sugar backbone, which is a deoxyribose or like a 5 carbon sugar and a phosphate group, right? Those two things attached to a nitrogenous base. So these are your nitrogenous bases, and these are basically your letters of the DNA, and these letters make a word, and that would be an amino acid. And these amino acids make a sentence, which is like a protein, and those proteins make a paragraph, which is like an organelle, which makes you know short story, book, and eventually like a human or some other animal. Um, the nitrogenous bases are A to T and C to G in DNA specifically. And there's this thing called 3' and 5'. Basically, 3' is when it has a sugar at the very end, and 5' is when there's a phosphate group at the very end. So when you have 3' to 5', you have what's called a coding strand, and that creates the leading strand in DNA replication. We'll get to that in a second. And when you have 5' to 3', you have a template strand that creates the lagging strand in DNA replication. So taking a look at the thing I just mentioned, this is DNA replication, and this looks really bad because it is. So you have this thing called the helicase, which is unzipping your DNA, and it splits it off into two things: your leading strand and your lagging strand. The leading strand is pretty simple. You have your DNA polymerase, and it is this thing that basically just grabs these hanging around nucleotides and just copies down the whole thing like this, right? And this is your leading strand. And it's the opposite of the strand that you had before. So you had 3' to 5' going in. This creates a 5' to 3'. So that it copies it that way. The lagging strand, on the other hand, has a much longer sequence. First, you have something called an RNA primer, which brings in RNA. After that, a DNA polymerase sees that and says, "Oh, I have to go copy some." So it creates some DNA in this like strand, and then it stops, and that is what you call an Okazaki fragment. Afterwards, DNA Pol 1 replaces the DNA with RNA, but there are still some gaps. And it's the DNA ligase that repairs those bonds and fixes the DNA so it looks good. The reason I think of it as like lagging strands is because there's so many more steps as like lagging behind to finish everything. But that's basically it. If it's a template strand or 5' to 3', then it makes lagging strands. It does this whole Okazaki thing. While if it's 3' to 5', it's a coding strand. It makes this leading strand right here, right, 5' to 3'. That is continuous compared to these fragments. So DNA will eventually have mutations. Okay, that's just one of the way things work. You have your mutations, right? So let's say this is my original, right? I have this right here. This is what it looks like. And it's just this. The replacement mutation would be, instead of there being like a little dot here, right? Or like an A, it would actually be a T, which is like a triangle like this. And this usually leads to like a silent mutation, which is when nothing happens, or a missense mutation, which is when one amino acid changes, right? And that's what like leads to more like healthy, you could say, like, or more gradual evolution, right? It's like these like small tweaks. Now you have insertion is when they add just like a new complete thing and it shifts everything to the right by one. Everything after that gets shifted to the right. And the complete opposite of that would be the deletion, where it takes one and everything gets shifted to the left. Everything to the right of that gets shifted to the left one. And this usually leads to nonsense mutations, which is when an early stop codon is created and the protein ends early. Now I have this thing right in here, but I'm not going to get to it yet. Instead, I'm going to talk about RNA transcription. So genes are just genes. They don't really do anything until you do transcription, which is basically the creation of this like message that gets sent to the ribosome, which gets turned into a protein through amino acids. So RNA transcription is pretty simple. Basically, an RNA polymerase goes through the DNA and it just copies things. Before it leaves the nucleus, there's two things that happen to it. First, RNA can have introns and introns and exons, and it gets spliced up in the nucleus. And introns are cut off and stay in the nucleus, while exons are put together and sent out. They exit the nucleus and become mRNA. But before they actually do that, there's one additional thing after splicing. There is the poly A tail, which is added to the 3' end, and a 5' cap put on the 5' end of the RNA. This is to allow the RNA to last longer in the cytoplasm because it's just eventually going to degrade as it goes on. So this is just the best way to keep it, working, is the idea. What is an exon and what is an intron can change depending on like environmental factors. So for this example, we have introns, extrons, introns, extrons, introns. So that could be when the animal is asleep. But when the organism is awake, it could be extrons, introns, extrons, introns, extrons, right? So just like completely flip that and make a completely different protein. And speaking of proteins, this mRNA is sent through the ribosome. The ribosome is made out of rRNA as well as some other things, and it is basically like the loading site for mRNA. So mRNA goes through, and it is read like those music boxes that you turn around. There's like those little dingly dangles that make the noises, depending on where it is. And instead of that, you have these things called tRNAs, which read the sheet music, and they come in and drop off a specific amino acid. They make a specific note. And this amino acid goes right here to the top, and it starts creating a sequence of proteins, a sequence of amino acids, which makes a protein, And after it drops it off, it just leaves and comes back to recharge and does the same thing. So you have a bunch of tRNAs coming in, replacing or putting in an amino acid, then leaving, and then doing the same thing. They're like taxi services, right? Except they know exactly what they're doing and where they're supposed to go and when they're supposed to come in. So it's more like Uber, if anything. So rRNA is the RNA that makes up ribosomes. There's some other things, but it's specifically rRNA that makes ribosomes. There's no other RNA that does that. And then, as I mentioned, tRNA or transfer RNA is the RNA that brings in the amino acids. Okay, now let's go back to transcription factors. So, if you want your transcription to happen and it's like copying stuff, it has to be read correctly. So you have your RNA polymerase, and it will have a sequence usually that is basically saying like, okay, start right here, right? I have mine right here as AGA. This is your promoter sequence. This happens specifically in like bacteria mostly. So it goes in and it's like, okay, I'm good to go. And it just shoots off and starts creating ribosome stuff. That would be a promoter, okay? That's a promoter sequence that's saying, okay, good to go. A repressor is something that basically covers the activation site, covers that activation sequence. And the RNA polymerase, it can't get through, it can't replicate the DNA, and for whatever reason, these things can be turned on and off, allowing for RNA transcription to happen or not to happen, depending on environmental factors. Like, let's say you need a milk-breaking enzyme, it's going to be off if there's no milk, but if there is milk, then it'll activate, it'll bond with that, it'll change shape, it'll let go, the thing will go through, and it will make the milk-breaking enzyme. So that's just one transcription factor. Another post-transcription factor that you have to worry about is this whole microRNA deal. Basically, when DNA is transcribed, you might have something called duplex RNA like this, where it has this weird bump where the DNA or the RNA doesn't match. And this gets cut up into mini RNA and this weird like bumpy RNA. And those are the two mRNAs. And they get put over this protein called the mini RNA protein complex, or the micro RNA protein complex. And if it's a perfect match, then it chops up and translates the RNA as per usual. If it is not partially matching, like with that weird bumpy one that I just mentioned before, there's no translation. So this basically reduces the expression of a specific protein in half is the idea. Let's see what else is that. Okay, post-transcriptional factors continued. There are some other things that could happen. Proteins, after they're made, can actually get chopped up to actually start working. Phosphate groups can be attached to the protein as well to activate it. And there's also something called ubiquitination, which is when ubiquitin attaches itself to a protein and basically says, "Hey, throw me away." That's the whole idea of it. Basically, when the the protein has done its job, it's retiring and it's taken off to the farm. Plasmids are a thing that will be brought up sometimes. It is a small circular piece of DNA. It's usually found in prokaryotes. It is double-stranded. It's not a part of the chromosomes of like regular DNA. And as I said, it's usually found in prokaryotes, but it's also sometimes found in eukaryotes. And it copies by itself. It doesn't need to replicate with everything else in like the nucleus. And it's not an essential gene carrier, but it does provide survival advantages at some times. That's the whole idea of it, I guess. Now, let's move on to a different thing. Bacteria CRISPR. So you have this little virus right here. A virus is a vector or a carrier of genetic material, and it wants to go into the DNA and basically say, "Hey, make more of me." And the DNA will go, "Okay." And the cell will start making more and more and more and more of this virus until it explodes, and now there's a bunch of viruses about doing their whole thing. Now instead, after it injects this genetic material, it could go into a specific section near this flank RNA or flank DNA. And when it is transcribed, you have this viral sequence, right? That's the virus sequence that was carried by the vector, and this flank RNA mixed with a little bit of tracer RNA, and that all binds to Cas9. And that is basically giving the cell instructions like, "Hey, if you see this virus right here, shoot it dead, Next time it comes by, get rid of it." And this is basically the same way how GMOs are made, where you have these like little viruses come in, inject a certain kind of DNA that makes strawberries sweeter or bigger or whatever, or more immune to pesticides, stuff like that. Okay, let's talk restriction enzymes. So restriction enzymes cut up DNA at specific coding sequences. It can be a sticky cut or a blunt cut. It is joined back together with DNA ligases, that's the whole idea of ligation. It is easier to fix sticky cuts than harder cuts, or it's easier to ligate sticky cuts than blunt cuts. That's the whole idea of that. And that's what it is. Next is epigenetics. Genes aren't the only thing that affect your traits. Epigenetic factors are things that attach to DNA and activate and deactivate a trait based on stimulus from the environment and usually stay for 4 generations. So to give an example, people have been doing diets for generations, right? People will not eat for days, not like all this stuff to become skinny. But what they do not know is that they are stressing out their body a lot, and they are basically telling it, "Hey, I'm running out of food, and I need you to help me retain energy, store it into fat for longer so I can live." So by trying to become skinny, you are making it easier to become fat, for lack of a better term. And not only does this affect you, but it affects the next 4 or the next 3, maybe if you include yourself as 4. But if you just write on your test, the next 4 generations of people. So you're going to be more likely to be fat. your kid is going to be, your grandkid, etc. Electrophoresis. Electrophoresis is when restriction enzymes cut up DNA and they make segments, right? That's the whole restriction enzymes thing. Based on the DNA sequence, you can find out whether a DNA is the same as, or a DNA sample is the same as another DNA sample. The closer it is to the well, as it's called, the longer and larger the segment is. So let's look at, for example, like let's say we're at a crime scene, right? And this is the DNA as found at the crime scene. This was suspect 1 and this was suspect 2. If the DNA is the same as the suspect, then once it's cut, it should have the exact same distance of cuts. And if you look at the crime scene and suspect 2, you can see that it's the exact same like distance away from this well at the bottom, compared to suspect 1, which is completely different. There's no match at all. And that's basically how electrophoresis works. That's how they tell if you're the one who done it or not. Actually, one thing I forgot to mention with CRISPR, no, but yes, I did. I totally mentioned that. It's how DNAs or how GMOs are done. Okay, cloning, embryonic stem cells. Embryonic stem cells is basically when you have your like an autosomal cell, like for example, a say you have yourself like a piece of your skin. You take out like a skin cell. You just take one out, and you have this fertilized egg cell. You pluck out the nucleus from that fertilized egg cell, and you take the body of your skin cell. You like after you rip out the nucleus, and you put the nucleus from the fertilized egg cell into the body of the autosomal cell, and you make a new zygote, and you zap it with electric charge to start the cell cycle, and it'll start to create a like a baby, like a fetus. Induced perpetuant factors induced perpetuant cloning is when you have yourself a cell that's already has its job, like, oh, I'm part of the skin, oh, I'm part of the heart, etc., and it's locked in what it's doing. Now, there are these things called Yamanaka factors, or you can just think of it as just like these factors that once you add these to the skin cell, it will become like a blank slate, okay? It's basically a stem cell all over again, and that gives it a chance to differentiate into like a neuron or a blood cell or something else. And that's the idea of cloning. You're cloning another like itty bitty piece of a cell. That's all that you're doing. With that being said, that is just about everything that you can expect to see for AP Bio, specifically for the DNA unit. Good luck on your tests. I have nothing else to say. Thank you for watching.